brief presentations, which are all related. We request that guys mute your mics. Okay, so we're going to look at MHC, transplantation, and blood transfusion. So the success of tissue and organ transplants depends on the donors and recipients' human leukocyte antigens. These are things you have gone through. I'm sure you know what HLA is, which is human leukocyte antigen. It is the synonym for MHC, which was found in rats. For the human beings, it was referred to as HLA, human leukocyte antigen, but it's basically the same thing. And these are called alloantigens, and they are found on the short arm of chromosome 6. And we all know that we have two major histocompatibility proteins, class 1 and class 2. These are coded by genes, as we already know, proteins are coded by genes. So class 1 is coded by HLA, A, B, and C. And HLD loci determines class 2. So class 1 is HLA, A, B, and C, and class 2 is D, which is also subdivided into DQ, DP, and DR. And then between the loci, there is a third class, which basically codes for complement. As we inherit this, each person has two types. You get one from the mother, and you get the other one from your father. And they are so much diverse. That's the word polymorphic. They are so diverse. As you can see there, HLA has 47, 88, and 29. And HLD, which is MHC2, has over 30 types or haplotypes, we may call them. But it's very important to note that each individual inherits only a single allele or a single type at each locus. So if it is HLA-A1, out of the 47, you will only have one. Out of the 88 for B, you will also have one. And out of the 29 for C, you will have only one. And among the HLA D300 genes, we have DP, D, QP, and R. So you'll have one P, one Q, and one R. The expression of these genes is said to be codominant because all of them get expressed. Whatever you have from your mother and your father all get expressed. So they are also what we call the minor antigens, as we've talked about major, but there's also minor antigens. As you see down there, we match for the major so as to prevent tissue rejection, but eventually we have tissue being rejected, and we think that the minor antigens do play a role in this rejection. These class 1 MHC proteins are found on all nucleated cells. That's one takeaway point for you. So MHC1 is found on all nucleated cells, all of them. And they are composed of a heavy chain and a small beta microglobulin. The setup or the structure of these MHC1 and MHC2 are similar to the antibody structure. So if you understand the antibody, you will understand the structure of the MHC. It's referred to as the immunoglobulin supergene family. It has the immunoglobulins and the MHCs. The class 2 proteins or MHC class 2 are found mainly on what we call antigen-presenting cells. The other one is on all nucleated cells, minus definitely the red blood cells. And then we have this MHC class 2 is present on all antigen-presenting cells, so what we call professional antigen-presenting cells, which include the macrophages, the B cells, and the dendritic cells. So the ability of T cells to recognize antigen is dependent on the association of the antigen with either MHC class 1 or class 2. At the end of this, you see what we've called MHC restriction, that these cells are able to see antigens that are carried by specific MHC. If it is a cytotoxic T cell, it will recognize antigens that are being carried by MHC or presented by MHC class 1. If it's a helper T cell, it is going to recognize antigens that are carried by MHC class 2 proteins. So that's what is referred to as MHC restriction. They will not recognize, cytotoxic T cells will not recognize an antigen that is carried by MHC class 2 and vice versa. These MHC proteins and genes, we have seen that they are important in antigen recognition, but they are also important in autoimmune diseases. They also play a role in autoimmune diseases. I don't know if you've already gone through autoimmune disease, but you will definitely go through it. It's basically the body fighting itself. That's what we call auto. The body is fighting itself. And these MHC genes do play a role in generating autoimmune diseases. They also play a big role in compatibility of donors and recipients. So number one, we have seen they play a role in autoimmune disease. They play a role in donor and recipient rejections. Okay, so that was a brief on MHC. Very importantly to understand that MHC 1 and 2, they are found on the short arm of chromosome 6, and MHC1 is present in all nucleated cells. MHC2 is present only in professional antigen-presenting cells. For example, the B cells, the macrophages, the dendritic cells. The MHC1 is, or HLA1, HLA has A, B, and C. That's MHC1. Okay, so tissue transplantation is a moving of a tissue from one individual to another, or from a donor site to another site. So it is basically you're either dealing with a site within the same individual or an organ from one individual to another individual. That's the classical definition of a transplantation. And it is aimed at replacing a damaged part or an organ or an absent part from an individual. And these are divided into 4 depending on the genetics of the individuals involved. We have what we call individual's own tissue from one side of the body to another side of the body. Common example is blood transfusion. If you're going for an elective operation, they can take off blood from you and keep it, and then after the operation, they give it back to you. Or the other one is a skin graft. They get skin from one part of your body and put it on another part of the body. The other one is called a syngeneic graft, which is basically a transfer of tissues between individuals who are genetically identical. Here we have identical twins. Of course, these are not very many because we don't have so many identical twins. The commonest other grafts are the allograft, which is basically a tissue transferred between two individuals of the same species. They are genetically different, but they are of the same species, as happens between humans and humans. The other one is a xenograft, which is a transfer of the tissue from one individual to another, but of different species. So autograft is same individual. Xenograft is individuals with the same genetic makeup, same species, same genetic makeup. Allograft is individuals of the same genetic makeup, different genetic makeup, but same species. And xenograft is different species. And as you will see, those are very easy to reject. And the rejections are basically also grouped into three. There's acute, hyperacute, or accelerated, and then chronic rejection. And these all depend on the other parts of the immune system. The acute rejection definitely shows that there are already preformed components of the immune system that actually play a role, similarly to hyperacute rejection. The chronic rejection comes on when involves components of the immune system that are built over a period of time. And definitely, we'll talk a bit about allografts, which are rejected. And we know that most of the grafts will definitely be rejected unless something is done. So eventually, most of the grafts get rejected. But we can be able to prolong that period of rejection by offering immunosuppressive therapy. So people who are going to be given grafts are also given immunosuppressive therapy, and some rejection is mediated by T cells. This is now to show that if you have another graft that is given to you after the first graft, this second one will be rejected very fast. So that's what we call the accelerated. And this primarily will depend on somebody who already has pre-sensitized cytotoxic T cells. So they do play a role in, those are the ones that will play the role in removing the graft. As you probably are already aware, foreign MHC proteins are more immunoreactive to T cells than foreign non-MHC protein. That is really just talking the same language. You know that the more foreign an antigen is, the higher the response to it. So that's just repeating some of the things which you already know. So that's hyperacute rejection. And this occurs within minutes of a graft being given. This is common in blood transfusion. People who receive blood that is not properly cross-matched, the reaction is immediate. And this is usually due to preformed antibodies. Chronic rejection, a graft that survives an acute allograft reaction can still become non-functional as a result of chronic rejection. And this takes months or even years. For many people who have received, for example, kidney transplants, 5 years or so, the graft gets rejected. And it's not a very nice story. The cause is unclear, but incompatibility of minor histocompatibility antigens and side effects of immunosuppressive therapy are said to play a role. And the main pathogenic finding in these people is basically atherosclerosis. The vessels get occluded, and eventually there is no blood supply to the donor organ, and it basically just gives way and dies. Okay, so we've looked at what transplantation is, what are the types of grafts, and what are the types of rejections. So if you can just reflect back to define what transplantation is, you can define the different types of grafts, and then the types of rejection of these grafts and the basis of the rejection. We will then look a little bit at how tissues are matched, because like in blood transfusion, we do what we call grouping. I'm sure each of you by now should have done their blood group, and you know you are either A plus or A minus, O plus or O minus, so that in case you need blood, then it's easier for people to get you blood. But when somebody requires blood, they send their sample to the lab, they group, and they say, okay, this is blood group A plus. They get the donated blood A plus, and then they have to match it. They have to mix it to be sure that it is not going to react when it is given to an individual. So that's the same process that is used here in HLA typing. This is tissues. The other one is more, this blood, liquid, these are tissues. So to determine the closest MHC match between the donor and the recipient, we do what we call HLA typing. And there are two methods that are commonly used to determine your haplotype, whether it's the class 1 and class 2 alleles on both the donor and the recipient chromosomes. The mechanisms involved and methods are quite complex. I wouldn't want you to waste a lot of time. We use molecular methods, which could include DNA sequencing and serologic assays. Just remember those two. So we use molecular methods, or we can use serologic assays. And these are other methods that you will read about if you are really an A student. Let me just get this away. Yeah, so there's as we do for blood, we also do matching. We don't only just do tissue typing, but we also do matching of these tissues. Okay, so that is tissue typing and tissue matching. Most important thing for you to know is that you match for all the MHC1 and MHC2, and you use different methods. There could be serological or molecular methods. There is always a question, because we know that the fetus is a foreign agent, and people ask, why is it not rejected? Why are others rejected and the fetus is not rejected? So why is the fetus not rejected? Yet it has foreign MHC antigens which are inherited from a father, even from many pregnancies of offsprings of the same MHC haplotypes. It's a unique thing because we know that it's a foreign antigen. It has foreign MHC antigens, but somehow it is not rejected. And the clear reason is that it's not clear. We are not sure why it is not rejected. There is just a possible explanation that the trophoblast layer of the placenta does not allow maternal T cells to enter into the fetus. That is the possible explanation. Okay, so when donor and recipient tissues are well matched by mixed lymphocyte culture or HLA typing, we ensure that we have long-term survival. So that's just to beef up what we just saw, that when you want, somebody mute yourself, please. So if we need survival of our organisms, we need proper matching. But there are also certain instances where tissues are not rejected, like the corneas. Corneas are never rejected. This is done by the ophthalmologists. They're easily grafted because they are avascular. There are no blood vessels in the cornea. That is why they are able to survive. But other tissues, as we have said, require lifelong immunosuppression. Although immunosuppressive doses. Somebody mute yourself, please. These doses keep reducing with time, and in some patients, a state of tolerance ensues, and drugs can be stopped altogether. But that's after a very long time. Yeah, we've already talked about this. You can read it a little bit in depth. There is immunology of rejection. We can give drugs like azathioprine, which inhibits DNA synthesis and therefore blocks growth of T cells. Okay, specific examples, we have the bone marrow transplant, which are really good for people who have lost their bone marrow, and these people eventually get to be cancerous, and these bone marrows are lost due to cancer, due to chemotherapy or radiotherapy, and you can be given a bone marrow to replenish your blood cells. One key problem with bone marrow transplants is called the graft versus host disease, where the graft is immunologically competent, okay? Immunologically competent donor cells, or the graft is immunologically competent and is given to an immunologically compromised recipient. So basically, the donor cells are more powerful than the recipient cells, and they therefore end up destroying them. That is what we call the graft versus host disease. It is common in bone marrow transplants. So that's basically what we have basically talked about, requirements for you to have a graft versus host reaction. You must have a graft that is immunocompetent and a host that is immunocompromised, and the recipients must express antigens foreign to the donor, and then you have a problem there. We have already talked about the graft versus host disease, which is caused by the reaction of the grafted mature T cells in the bone marrow inoculum with alloantigens to the host. It could be acute or could be chronic. We have we have heart transplants, rejection fractions of up to 20%, and life expectancy is less than 1 year. So even with a heart transplant, you won't live long. First heart transplant was conducted in Africa in the University of Cape Town. Yeah, so that is it. The survival is 80% at 5 years, but at 5 years, 50% of these people already have coronary vascular disease. So you remember what we talked about in chronic rejection. The kidney, this is one of the commonest tissues that are being transplanted. Even in Uganda, soon we shall have kidney transplants being done in Mulago. They're quite common. You've heard of people stealing kidneys and selling kidneys. It's a big business, and most of these kidneys are blown by hypertension and diabetes. So if you are still young, please make sure you don't get any of these diseases. They'll blow up your kidney. And kidney transplant will save you some money, which is usually high cost of renal dialysis. So if you do a transplant, you'll be able to save some money. Liver transplants, they said 1 year survival exceeds over 75%, and 5 year survival is 70%. So transplantation works. Okay, so that's been transplantation. We are going to quickly run through blood grouping. Janan, please, can you put on your shirt? Blood grouping. Transfusion is the transfer of blood or blood components from one individual to another. That is something which you already know. We've already talked about it. Infusion is the introduction of fluid other than blood, for example, normal saline or glucose. The blood group is determined by the antigens, that is the agglutinogens on the surface of red blood cells. Antibodies or agglutinins can bind to red blood cells antigens, resulting in agglutination. So that is, you must have done a class on antigen antibody reactions. So we're just describing agglutinins, agglutinogens, agglutination. The common blood groups that we have the ABO system, we have the rhesus system, plus others, including the Duffy and the Whale systems. But commonly, we look at the ABO and the rhesus. Like we saw tissue typing, we also do typing for blood. And if somebody is blood type A, means he has antigen A and antibody anti-B. So if you're blood group A, you have A antigen and anti-B antibody in your blood. If you're blood group B, you have blood group B and antibody A in your blood. If you are blood group AB, means you have antigen AB and no antibody. If you're blood group O, you like myself, you neither have antigen A or antigen B, but you have anti-A and anti-B. So this is just to show you what we've been talking about. Antigen A and anti-B, antigen B and anti-A in your plasma, antigens A and B, and neither anti-A nor anti-B antibodies in your plasma. Neither, if you're blood group O, you neither have antigen A nor antigen B, but you have anti-A and anti-B antibodies in your plasma. So when we are doing transfusion, as I told you, we do what we call grouping and cross-matching. Grouping is just to know your blood group, and cross-matching is to see that your blood does not actually react with the donor blood. If the wrong blood type is used, the person's own immune system immediately attacks the donor's blood and causes clots and red blood cell destruction that can lead to total kidney failure and death. That's very important for us to ensure that we have done good cross-matching. Okay? So as you see up there, most people are blood group O, which is good because then they can give to everybody. A person with blood type A can receive blood from a donor with blood type A only. Okay? If you are blood group A, you can only receive blood from blood group A only. The anti-B antibodies in the recipient do not combine with the type A antigens on the blood cells of the donor. That's how you're able to receive blood. If you're blood type A, you have anti-B in your blood. So when you are given A, the B cannot react with the B. A person with blood type B cannot receive blood from a donor with blood type A. The anti-A antibodies in the recipient will combine with the type B antigens in the red blood cells of the donor. Okay, then we have rhesus factor. Rhesus is basically a monkey, so that's where this was discovered. And we have individuals who are positive or negative, who either have or do not have this rhesus factor. So if you have it, you'll be rhesus positive. If you don't have it, you'll have rhesus negative. Anti-rhesus antibodies of the system are not normally present in the plasma, but can be produced if an individual with rhesus negative is exposed to rhesus positive blood. There's what we call hemolytic disease of the newborn, which is very important for you to understand. A small quantity of blood, of fetal blood, leaks across the placenta into the maternal blood system. If the mother is rhesus negative and the baby is rhesus positive, the mother's immune system begins to produce anti-rhesus antibodies. So this rhesus positive blood leaks into the mother's blood, and the mother forms antibodies to rhesus. That's what you just need to pick away from this. The mother's antibodies then cross the placenta during the next pregnancy into the fetal blood. And if the second fetus is also rhesus positive, the antigen-antibody reaction causes hemolysis of the fetal red blood cells, and this results in what we call hemolytic disease of the newborn. So this is a schematic presentation. You have a rhesus negative mother who gets pregnant, impregnated by rhesus positive father. The baby is rhesus positive. The rhesus positive antibodies will cross into the mother, and when they cross the placenta, the rhesus positive antigens cross the placenta to the mother to form rhesus positive antibodies. So in the first pregnancy, nothing happens. There is no problem. It's just that the rhesus positive antigens cross the placenta to the mother's system. Mother forms rhesus antibodies. In the second pregnancy, when the mother carries a rhesus positive baby, these antibodies cross over to the baby and destroy the red blood cells of that baby, which is really very bad. So all hope is not lost. I would encourage all our girls and our boys to test their blood group and know which blood group they belong to. Because it's very important, if you're a woman and you get pregnant and you're rhesus negative, the risk of losing a baby in the next pregnancy is very high if you get a rhesus positive father. So please try to check your blood group. They will tell you whether you're rhesus positive or rhesus negative. So if a woman is rhesus negative and gives birth to a child, or if she has a miscarriage or abortion, she's given an injection called anti-RH antibodies. So they give you anti-D, as it is called, when you get pregnant. The antibodies bind to the fetal rhesus antigens and inactivate them if they cross the placenta during birth, and the mother's immune system does not respond by producing antibodies. I think that should be the end of my presentation. I welcome questions.
Excuse me, Doctor.
Sir.
Yes. You can ask.
Excuse me, sir.
Yes.
Thank you for the presentation, but according to the rhesus compatibility,
Yes.
on the mother and the father, who determines the rhesus type of the baby?
Most times it's the father.
Okay.
Yeah.
Excuse me, sir.
Yes.
My question is almost related to that, but I'm wondering if the mother is positive, and can't we have the fetus being negative?
It is a possibility. It is a possibility if you marry a rhesus negative husband.
So the same thing happens.
The same thing happens whether if the mother is negative and is carrying a rhesus positive baby, or if the mother is positive and carrying a rhesus negative baby. The issue is that the rhesus antigens cross over and then antibodies are made to them, and then you'll have problems in the next pregnancy.
Okay, then the last question, what is the best timing for giving anti-D?
Anti-D, I think, is given before the next pregnancy.
Thank you, Professor.
Yeah.
Excuse me, Professor.
Yes.
You say that a rhesus negative mother forms antibodies against the rhesus positive child. That's why we can have the fetus die during the course of the next pregnancy. Now I am wondering, will the fetus who is rhesus negative form the antibodies against the rhesus positive mother?
What?
At first you said if a mother is pregnant and a mother is rhesus negative and the fetus is rhesus positive, the antigens will cross through the placenta to the mother's blood, and the mother forms antibodies against it.
Yes.
So now I am wondering if it's the other way around. Will also the fetus form the antigens? Will also the fetus form the antibodies against that of the mother?
You see, it is these antigens crossing,
Yes.
And Yes. So if the mother is positive, the antigens cross to the baby, the risk is very, very low for that baby to die. Usually you need preformed antibodies. So in the first scenario, when the baby is positive and the mother is negative, you have antibodies that are made waiting for the next pregnancy. So the risk in the opposite is very, very low because there are no antibodies waiting for the next pregnancy. The uterus is always cleaned.
Okay.
Mm.
So should we say that in the second case, when the child is when the mother is positive and the child is negative, it is not harmful?
Yeah, that is it. The risk is very low. The risk is high when you have a rhesus negative mother and a rhesus positive baby.
All right, thank you.
Mm.
Excuse me, Professor.
Professor,
Yeah, yes.
I think maybe to add some voice to what you've told our colleague. The development of antibodies against maybe rhesus positive by a rhesus negative mother. It is the first exposure of a rhesus negative baby brings about production of antibodies, and these antibodies are going to wait for the next pregnancy. But if at all maybe the rhesus antigens from the mother cross to the baby, then yes, the baby may get antibodies, but the baby may never get a chance to get exposure to the mother's rhesus antigen. Therefore, the mother will not have challenges from the baby, but the next pregnancy, the baby is going to get attacked by the antibodies from the mother. Thank you so much.
Okay, thank you.
Excuse me, Professor.
Yes.
Thank you for the lecture. I'm wondering, what is the basis of saying that blood group O is a universal donor? Yet we understand from your lecture that blood group O has A antibodies as well as B antibodies. So in that case, if someone is given blood group A, if someone who is blood group A is given blood group O, that means that the antibodies that are present in the donor will react with A antigens that are present in the recipient. So what is the basis of saying that blood group O is a universal donor? Thank you, Professor.
Okay. So what do you think? You first let me know what you think.
What I think is that
Excuse me,
we shouldn't take it lightly that O is a universal donor. What we should do is, if, for example, we receive a patient, or if the patient is blood group A, we should first give them blood group A if it's available. If it's blood group B, we give them blood group B if it's available. Then if they are AB, we give them AB blood group if it's available. Then we resort to blood group O, the last resort, even after doing cross match.
Yes.
And we have confirmed that the blood will not agglutinate afterwards. Thank you.
Okay. Somebody else wanted to add something?
Yeah, I think blood group O does not have antigens. That's why we call it a universal donor.
Yes. That is very true, but as your colleague has submitted, it's always important. If it's A, give A. If it's B, give B. If it's AB, give AB. We should give O when we don't have the others, because there is always a risk somewhere.
Okay, excuse me, sir.
Yes.
Now, what if we have a case whereby a mother is having twins, and one of the twins is positive and another one is rhesus negative, but the mother is rhesus negative. So how can they go about that? And it's the second pregnancy.
Of course, if it is the first pregnancy, as we said, that's why we need to do rhesus blood grouping. If you discover that you are R negative and your husband is R positive, you have to take precaution and take the anti-D, before the next pregnancy. That's the only way out. Because there are many scenarios that could come in there. So it is always good to take precaution.
Yeah, now we are looking at this circumstance. Someone is in the village, she doesn't know about the rhesus things. Now here presents
the risk of one baby dying is high. Okay, the risk of one baby dying, if you are RH positive and your mother is negative, the risk of that one baby dying is quite high on the second pregnancy.
Okay, thank you.
Excuse me, Professor.
Yeah.
But I also, just before I make my submission to the last question, the person has posed, to reduce such cases, that's why the government, for example, in our case, is trying to ensure that antenatal care services are ready and as close to the catchment population as possible. So it's very rare that, you can go through antenatal care and that a bit of test is not run. But then I've also come across a few cases, Professor, whereby the mother is isoimmunized in the first pregnancy, but then in the subsequent pregnancy, the mother falls under a category. I've had the clinicians referring to it as slow antibody formers, whereby even the second pregnancy pass without necessarily receiving the anti-D. It may be risky to take a chance and try to see if you're in that particular category, but I've seen mothers who are isoimmunized, but in the subsequent pregnancy, they didn't get the antigen.
They come through.
Yeah, they come through without the baby presenting with the hemolytic disease of the fetus and the newborn.
Yeah, these possibilities are there. As we have said, diseases don't read books, conditions don't read books. The classical is to say, if you are positive, your wife is negative, and most likely you will produce a positive baby. So do the needful. In case you are one of those lucky ones who fall in the outliers, you can survive with it. And I'm sure there could be a number of people who have survived through it. But it's not worth taking that risk. It's better that you do the needful, you do the right thing.
Excuse me, Professor.
Yes.
Someone previously asked a question and they said a father is the one that determines the rhesus status. So my question is, does it for Nigerians that way of distribution, or every child who
does it what?
Do siblings of the same father have the same rhesus? Children with the same father, do they share?
Yeah. The rhesus factor is not as well understood as all the others. So it is not very clear if all the children will have the same rhesus or some will have negative or positive. So it's not very, very clear how that really happens.
Excuse me, Professor.
Yes.
Assuming a mother is rhesus positive, then the fetus is negative. Then we have some crossing to the fetus, and the fetus is able to develop antibodies against the antigens. And that fetus in our case is a lady. So that in future there is a possibility of her getting pregnant, and those antibodies developed maybe affect the pregnancy.
Okay. Sorry, I didn't get your question. My network jumped.
I said, assuming we have a rhesus positive fetus in a rhesus negative mother. So sorry, a rhesus negative, rhesus positive mother, then the fetus is negative.
Okay.
Blood occurs and the fetus develops the antibodies. But then that fetus is a lady in our case. So can those antibodies developed in that time stay viable enough up to when a fetus maybe reaches the stage of giving birth herself and affect
producing?
Yeah. And also the antibodies
I wouldn't
affect.
think that. Okay, let me see if I try to get your question. You said you have a baby who is rhesus negative, produced by a rhesus positive mother, and somehow the baby is born. Will those antibodies stay with this lady until she's grown and destroy the next baby she carries?
That was
Is
the
that
question.
the question? Yeah, I don't really think so.
Maybe another one. You say in bone marrow transplant.
Yes.
First of all, we see that our immune system develops over time.
Yes.
So, maybe due to immunization and also the pathogens you get exposed to in your lifetime. So during the bone marrow transplant, do they have to coach those grafts before they are maybe implanted into the recipient?
Do they have to do what?
Different immunogens. Do they have to coach the grafts that are transporting to the recipient to develop immunity against the different pathogens?
Are you getting me?
Yes.
Hello? Yeah. Yeah. The question is, do they have to coach the different immune systems?
Do they have to coach the grafts to develop immunity against the different pathogens that maybe the recipient is most likely to get exposed to before they are actually put into the recipient?
No, they don't.
Excuse me,
so they don't. They just actually what they do is they try to make sure that the donor tissue is not stronger than the recipient tissue. That's why they give you immunosuppressive therapy to try and tone down the donor's immune system so that the donor tissue doesn't fight the recipient tissue. Okay. I think this will be the last question. Yes.
With patients having graft versus host reaction, they usually present with jaundice, a sign of hemolysis. What is the why is there hemolysis with the graft versus host reaction?
There is destruction of You said there is jaundice in graft versus host disease. So if it's blood cells definitely that are destroyed, that's how you develop jaundice. You have cells being destroyed. That's how you come up to develop jaundice.
Excuse me, Professor.
Yes.
Why is it that blood group O is considered the strongest, yet it can predispose you to some diseases like ulcers?
That's very interesting. I don't think it is considered the strongest. It's the most common. It's the most people are blood group O. So it's not that it is the strongest. Okay, it is just that many people are blood group O. So people tend to think that if your blood group or predisposition to diseases, there's not a very, very clear linkage between blood groups and diseases. Sometimes people may find some relationships, others there's a correlation. There's no clear-cut distinction. Say if you're blood group O, then you are likely to have ulcers. Okay, thank you so much. Have a nice day.
Thank you, Professor. Thank you so much. I'm sorry I logged in a bit late.